Our next speaker is Dr. Leila Pirhaji, founder and CEO of ReviveMed. Welcome to the stage, Leila.
Doctor, my eyes have seen the years and the slow parade of fears without crying. Now I want to understand.
Hello. Hi. I would like to thank the organizer for inviting me and the opportunity to be here. And I'm really excited to tell you guys that how we can be in the edge of drug discovery and development and how we could push the boundaries. In 2003, when we sequenced the human genome, we thought we would have the answer to treat many diseases. But the reality is really far from that. And there are thousands of diseases with no treatment. Because in addition to our genetics, there are many other factors that lead to a disease. And there are only less than 10% of diseases with a strong genetic cause. So one of the biggest things is our lifestyle and diet and environment. You guys have probably heard a lot how stress, smoking, exposure to toxin, and unhealthy lifestyle lead to developing many major diseases. In addition to that, we've been hearing more and more about the role of gut microbiome in our health. These are the bacteria that live in our gut, and the changes that happen in our gut microbiome have linked to several diseases, inflammatory diseases, mental disorders, even depression, and even cancer. So our biology is incredibly complex. There are all these different factors from our genetics and environment and our bacteria in our body leads to developing diseases. So in order to be able to develop effective therapeutics, we need to understand all these different elements and how they interact with each other, and then we are able to develop effective treatments. This lack of understanding of biology has been one of the major failures for drug discovery and development. Pharmaceutical industries start by screening thousands of compounds to hit a therapeutic target in different in vitro models. And without knowing exactly what led to the disease, these drugs by serendipity move to clinic, and the majority fail. One of the examples that we have here is the case of Alzheimer's disease. You guys all probably know about Alzheimer's disease. It's a huge problem right now, and it has no treatment. It's a neurodegenerative disorder, and usually patients lose their memory and cognitive function. At some point, they are not able to take care of themselves anymore. Alzheimer's is really complex disease. We know that there is genetic risk factor associated to Alzheimer's disease. Our diet and lifestyle has a significant role in developing the disease. Patients with obesity and diabetes are in increased risk. Some people call Alzheimer's the diabetes of the brain, because actually if we eat more sugar, it actually increases our risk of developing Alzheimer's disease. And sadly, because of all this complexity and without knowing what exactly caused the disease, there has been many attempts to develop drugs that help Alzheimer's patients, but everything has failed so far. And this long line of failures actually has been one of the barriers, because right now the pharmaceutical companies have shut down their Alzheimer's programs or neurodegenerative programs. So we need to understand biology better to be able to address all these problems. And this is one of the things that we hear. This is the ugly trends of pharmaceutical industry. Right now we are spending more and more money and more time on discovering therapeutics, and we are developing less and less drugs. Pharmaceutical industries have been one of those industries that the efficiency has decreased by time. 30 years ago, it only cost $200 million to develop a drug. Right now, it costs over $2 billion, about 15 years, and 10% chance of success. So we are in a desperate need of innovation and innovative technologies to improve this efficiency. And even in a small amount of improvement, it could have significant saving of time and cost. And most importantly, for patients, because there are many, many patients with no treatment. So what can help? Now, we've been hearing a lot about artificial intelligence and how artificial intelligence, or AI, has been disrupting many different industries, from automation, transportation, finance. So how about AI? But AI is actually not new. The concept of artificial intelligence have been developed since 1950s, that Alan Turing bring up the concept of machine intelligence. And AI was born in a conference that happened in 1956 at Dartmouth, with the leadings of AI at that moment get together. But as we hear in the previous talks, computers back then couldn't save memory. And it was very hard to have access to computing powers. They were extremely expensive. So AI at that time couldn't live up the hype. And that led to two AI winters, which happened between 1970s to mid-1990s. It was until 1997 that IBM Deep Blue beat one of the world's champions in chess that actually AI got more tractions and people are starting more developing AI technologies. And it wasn't too long ago that the most advanced AI algorithm has developed. It was until 2010s that we saw that, like in 2011 was IBM that beat the champions in geometry, and 2016 was Google AlphaGo beat champions of Go players. So right now there is more and more interest in artificial intelligence. So can we use AI now for drug discovery? In fact, pharma has been one of the late adopters of artificial intelligence. But we've been seeing the trends. For example, the number of publications that happen using AI in drug discovery since 2010 have significantly grown and has been more than any prior year of that beforehand. We've been seeing more and more partnerships of AI startups with big pharmaceutical companies. Big tech gigantic companies, now they're moving this space, like Google and even IBM. They're starting these healthcare and pharma units. And there are even tech investors that they've been starting more bio funds, that they were investing in the startup companies that use technology-based bio platforms, which all these players have been shaping the market. But where we can use artificial intelligence? Right now, we have been seeing that AI is being used in finding therapeutic targets, lead optimization, medicinal chemistry, and even all the way to finding right patients in clinical trials and optimizing clinical trials. But I think one of the most important roles of AI would be help us to understand biology, to understand this complexity of our biological system, that what really happened in a disease that we are observing the symptoms, and how we could treat those patients with that. So I've been always curious about medicine, and I always wanted to help patients. But growing up in Iran, my parents really wanted to become a doctor, a medical doctor, and not a PhD doctor, like many other parents in Iran. So 15 years ago, I went to medical school, even though I was really passionate about studying mathematics, and I was really good at it. Soon after, I left medical school because I missed mathematics. I think my parents were very disappointed at that moment. I changed my major to a field called biotechnology, which was recently established in the University of Tehran. And during my undergrad degree, I learned that I could use actually mathematics and computer science to study medicine. For me, that was eye-opening. I was like, wow, this is so cool. So since then, I've been developing algorithms to analyze biological data. But back then, AI wasn't as cool, specifically not in Iran. And it was a time that we just sequenced the human genome. And so I started my work by analyzing the sequencing data of even genome of E. coli, which was really small. But I was more passionate about how can I understand human biology? Can we collect data from all the molecules that exist in human body and then develop models to describe how they're dysregulated in a disease? And if we know those mechanisms, we could target that to treat those patients. So for doing that and reaching that, I came to MIT to get my PhD in this subject. But again, we faced that complexity. We were looking at all this data that we have from a human biology and from a disease. And we were looking at the genomics data, and we were saying, this is not enough. If we're looking only at sequence of genetics, we are missing all these other informations that lead to developing a disease. So we needed to have more data. We needed to collect more data about our biology to understand the details. But the good news was that in addition to our DNA, there are so many other molecules in human body. In fact, there are over 100,000 metabolites. Metabolites are any molecules that are super small in their size. Known examples are glucose, cholesterol, fats, things we hear all the time in health and diet. These molecules are very important, and they provide the most functional information about diseases because they are downstream of DNA, and they carry information about our genetics. They are also involved in our metabolism, so they could show lifestyle and environmental effects. And also, the bacteria in our gut interact with our human host through these small molecules or metabolites. So it's really important to understand these molecules. So for me, and also, when they dysregulate, they lead to many different diseases. For example, diabetes is actually one of the diseases that we know that the increased level of glucose leads to diabetes. It leads to cardiovascular problems. It could do many liver diseases. So it's very important to understand these metabolites to be able to develop effective treatments. And at that moment, during my PhD, it sounded straightforward. Okay, we're going to collect lots of data and measure all these metabolites for each patient, and then build our AI models and mathematical models to see how they're changed. But then again, I realized why no one has done this before. It wasn't that simple. And what we ended up was with this dark matter of metabolomics. We know these small molecules should be in human body, but we weren't able to characterize them in the lab. Because there are so many of them in each patient. There are over 100,000 of these metabolites, and each one of them is different from the other one. If I want to compare that to our sequencing our genome, in our DNA, we have these 4 building blocks of A, T, C, and G. So we run one experiment and sequence the whole human genome. But if we have 100,000 different types of molecules, it requires lots of customized experimentations to extract them from human body and characterize them. And some of these molecules, the fats, they solve in the water. Some of them are polar molecules like carbs, so they don't solve in the water. So it's really challenging. And another big challenge was that we could measure the molecular mass of some of these molecules, but there could be 10 molecules with the exact same mass, so we cannot figure out exactly what they are. The other challenge was that some of those molecules are formed in the cell, they activate a really important signaling pathway, and they're used very quickly. So they are present in the cell for a very short amount of time that we are not able actually to measure them. So all these experimental challenges together, current technologies can characterize less than 5% of human metabolites. And when we have such a little data numbers, we don't know what's happening. And I usually tell people, with that few hundred molecules that current platform can characterize, we don't need AI. You could just use Excel and figure out what happened. So actually, experimentally, if you want to solve this problem, it's estimated to cost billions of dollars and decades of research. So what we realized, experimental solution is not a solution. And we said, okay, can we actually leverage our AI platform to overcome this problem? And right now, we are in the right time in the history to be able to do that. Because now we have lots of computing power available with reduced cost. And also, there are exponentially growing biological data available in the literature to be able to use and leverage to overcome these difficulties. So what we did was, okay, let's build this database of current knowledge about these molecules and then use our AI algorithm to mine all this data. So what we did first, actually we went back and said, okay, whatever knowledge available about these molecules in the literature, what the chemical composition of these molecules could be. Are they detected in different tissues? Are they detected in our urine or our blood or even a stool? Some of them could be food metabolites, like vitamins. They are important metabolites in our body, which we rely on to absorb them from food. And when they're also dysregulated, they lead to many diseases. So we combine all this information about any of these molecules. But then we wanted to see also how those molecules interact, or metabolites interact with other molecules. So we combine information, interactions of metabolites to proteins, the interaction of proteins to other proteins, the drugs that are targeting these proteins, the association of drugs to diseases, the association of genes to diseases, the association of metabolites to diseases. Anything you may imagine in the literature, we combine them together. And the way that we combine them as this mega network. Each one of these nodes in this network represents a metabolite or a protein or a drug or a disease. And the edges in this graph also represent their connections, or if they interact or they're associated with each other. This network has millions of interactions, and then we built AI model to actually mine this network and give us therapeutic insight. Then what we do, we actually work with patients, we work with hospitals, we collect samples from patients like blood or tissue biopsies. And then we run these assays to measure the mass of a metabolite, the molecular masses of metabolites. But as I mentioned to you guys earlier, we don't know exactly what they are. And I have a very dummy example here. A molecular mass of 180 could be their glucose, galactose, or fructose. They all have exact same molecular mass, but different functions in our body. So current platform has to rely on doing more experiments to figure out this ambiguity. And of course, more experiments require more cost, time, and we can't do it for everything. What we said is that, okay, our AI algorithm consider all those ambiguities. It then mine that mega network that we created before and try to figure out how those metabolite masses that are changed in a disease are connected to each other. And because of the way that they are connected to each other, we are able to see, okay, this 180 is most likely to be glucose, because glucose is better connected to the rest of the things that are changed or dysregulated in a disease. So also, being able to understand why things are changed in a disease gives us novel therapeutic insight. Because now we know the details of molecular mechanisms, and we could go back and target some of those proteins that are causing those disease mechanisms. And some other existing compounds or drugs actually be part of those mechanisms, that we could use those drugs for new therapeutic effects. And then we validated all these findings. We were able to find novel disease mechanisms, novel therapeutic targets. We went back to the lab. I don't have the data here, but we validated everything. We published them in Nature Methods and Nature Communications. And this was the work we did at MIT. Moving forward, we also did more validations with the collaboration that we are doing right now with pharmaceutical companies. But we didn't want to stop there, because our goal wasn't to just develop the coolest technology. We want to change the world, and we want to save patients' lives. So we started ReviveMed, a startup company, to bridge this gap of academic science and the real-world impact. Our AI platform, we have developed this automated drug discovery engine that could significantly reduce the cost and time of drug discovery and development. Right now, just pharmaceutical companies spend hundreds of millions of dollars and years to develop it, to find the drugs before doing any testing in humans, so which is called like preclinical discovery. Our goal is to do this process is only 1 year and $1 million in spending. And because we are using data from those small molecules, that has not been possible before. Our drugs will have a higher chance of success rate because we are discovering the details of molecular mechanisms. Right now, my team is tackling one of the most important diseases that affecting our population, and metabolites are key drivers. As you guys may know, the rate of obesity and diabetes have been increased significantly in past years. According to World Health Organization, diabetes is going to be the number 7 leading cause of death by 2030. But many different diabetes patients develop different types of complications. Some diabetes patients develop liver problems. Some diabetes patients get cardiovascular problems. Some diabetes patients develop kidney issues. Right now, there is no data-driven way to predict which one of these patients will develop which type of complications and how to treat those patients. One of the first diseases that we are working on is called fatty liver disease or non-alcoholic fatty liver disease. This is one of the huge epidemic, and it's caused by accumulation of fats in the liver, mostly due to obesity or diabetes. There is a significant need for patients right now. We tackle these indications as a first disease because we have a unique way of collecting more data about those fats that accumulate in the liver. So we are able to understand novel mechanisms of the disease and novel approaches to treat these patients. Fatty liver, it's a very huge epidemic. It's right now affecting over 100 million of Americans in the United States and about 20 to 30% of global population. It has no pharmaceutical treatment, and when the diseases progress in the different stages, it leads to liver failure and liver cancer. And it's becoming the #1 reason for the liver transplant in the US. My team so far has developed this gigantic database and a comprehensive database about fatty liver disease and the comorbidity of it happen with it. So I showed you guys this gigantic network earlier, right? This is the part, it's only specific to our fatty liver disease. We figure out the genes, metabolites, and other diseases that are connected to each other, and the mechanisms, a novel mechanism that leads to connection of all those things. We have all, in addition to that, we are collaborating with leading hospitals in the US and also Europe, and we are collecting metabolomics data from hundreds of patients that have been diagnosed with the advanced stage of fatty liver disease. And we have already identified novel therapeutic targets and mechanisms that are associated with fatty liver program. We are right now in the process of doing more validation studies of our findings, and soon we are going to do preclinical validation in a mouse model of fatty liver disease as well. And fatty liver disease is just one example. Moving forward, we are going to tackle hundreds of diseases with no treatment. And by collecting more data about metabolites and understanding how changes in metabolites leads to developing different diseases, our algorithms will get smarter and smarter to develop the right therapeutics for the right patients. And we will get closer to reach our vision of saving lives with every line of code. Thank you.